The structure of antibody. Antibodies. Now, the structure of antibody in a simple and technical form is just a Y shape. But if you look deeper, you understand that the Y arm of the antibody consists of the light chain and the heavy chain from the down, and it is fragmented. It is a binding site of the antibody. Now, the immunoglobulins are located in the Y arm of the antibody, and you can see there is a bond that joins them between the light chain and the heavy chain, which is considered the disulfide bond. So this collectively jointed the heavy chain and the light chain in a very straight form. Whereas down there in the lengthy part of it, under the heavy chain, you can see we have the variable region at the top in the Y arm, whereas the variable region is the paratope, where it bonded with the epitope on the surface of an antigen. Then there is a constant region which never changes. It is close to the disulfide bond. Then there is the hinge. The hinge is the disulfide bond that joined them in the Y arm and also in the heavy chain region. So this is considered something very straightforward and technical, not more of a microbiology, but the aspect of the immunobiology to just understand the structure of the antibody. Now, for us to explain the different types or classes of immunoglobulins, we have to keep it in mind that all these immunoglobulins and their subset, they are located on the surface of the antibodies. They are not antibodies themselves. So there is difference between antibodies and immunoglobulins. Immunoglobulins, they are the main binding site on the surface of the antibody. So they are not the same. So we have 5 types of immunoglobulin. We have the IgA, IgG, IgM, IgE, and IgD. But collectively, for you to remember it, the formula is just you remember rearranging G, A, M, E, and D, which is gamete or gamete, whichever you call it. And as you can see, the IgG is a monomer. In chemistry, wherever you heard of monomer, it means one, a single unit. So it's one of the most abundant immunoglobulin on the surface of the antibodies, and you can find it in the blood. It has just a Y shape, with it fragment the light chain and the heavy chain, the constant region, the hinge chain, and the other aspect of it. So it is located in the blood and is one of the most abundant antibody that you can find in the blood. Then we have the IgA. This is a dimer. It is found in secretions like the saliva, the tears, the breast milk. That's why it is participated in the passive immunity, the transfer of immunity from mother to child either via umbilical cord or via breastfeeding. So it has high concentration, the IgE, and most importantly is that it composes so many secretory substances, as you can see from the demonstration. Now, number 3 is the largest antibody immunoglobulin that you can have, which is the IgM. The IgM is a pentamer. From chemistry, penta means 5. So it has a distribution of 5 collective monomers that comes together to produce a larger immunoglobulin. So it is usually when a B cell differentiated from stem B cell into memory cell, the first fragment that it produces is the IgM. So it's the first responder. It's like where there is an armed robber and the first individual that will arrive as a security agent will be the IgM. So it's the most largest and most effective. And it is first that it is differentiated after the production of the B cell. So you can find it uh, in a lot of infections. So when there is an infection and you test the presence of the antibody, you are going to find the IgM there. Then number 4 is the IgA. The IgE is also a monomer. That means it's just a one particular immunoglobulin, but it is responsible wherever there is an allergy. So when you are coughing or when you are scratching your skin, or when there is pruritus or urticaria or there is inflammation in the body, that means the first responder there will be IgE, because it is mainly meant for hypersensitivity and allergy, and it is located in the tissue. That's why it is effective in fighting parasitic infection. And number 5 is the IgD. The IgD is also a monomer. It is found on the surface of the B cell. So that's why it is a superficial immunoglobulin. And wherever you see a pre-B cell or a memory B cell, there is a IgD immunoglobulin. Then the concept behind the structure of antigen. The most important part of every antigen is that it has an epitope. An epitope is just a receptor site where the antibody can bind with it. When there is no binding, there is no antigen-antibody reaction. That's why the epitope is the most important part of the antigen. However, certain antigens, they can be antigenic, but they cannot be immunogenic, meaning they can be foreign, they can cause infection, but they cannot trigger immune response because the molecular mass is not enough and they don't have molecular complexity. So in such kind of situation, if it is in the case of vaccine, we use what is called the hapten. Hapten are not antigenic, but they are immunogenic. So when you combine an antigen which is antigenic with a hapten which is immunogenic, it becomes a very strong antigen that can trigger immune response. So that's why you can see a hapten here with a green color. And by the left, you see how this antibody is binded to the surface of the antigen through the receptor site. And the properties of antigen. For every microorganism to become a complete and perfect antigen, which is antigenic and immunogenic, it have to possess the following. First of all is foreigners. Since these immunogenic cells are located inside the human body and some of them in the peripheral blood circulation, then an antigen can only be recognized as an antigen when it enters into the human body. Anything outside the human body is no longer an antigen for the body. But immediately it enters the body, it becomes foreign to the body. So it can become an antigen. Number 2 is like molecular size. The higher the immunogenicity, the higher the molecular complexity. So anything above 100,000 Dalton, it is highly immunogenic and also antigenic. But anything below 100,000 Dalton, it is poor immunogenic and it may not be even antigenic. So that's why we have a hapten, which is antigenic but not immunogenic. And number 3 is chemical complexity. How complex it is physically and chemically. So you see that some certain antigens, they have so many spikes, so many distribution of pseudopod in such a way that it's very complex. It's not smooth like a cycle. It has a lot of other receptor site. So physically it is antigenic. Some of them, they have chemical complexity because they have high concentration of other negative substances. If it is a poison or a concentrated chemical, so it is chemically complex and therefore it is immunogenic. Then there is specificity, how specific, unique, and peculiar it is. That's the binding site. So an antigen have to have specificity in binding site. So long as the site cannot be binded with an antibody, then therefore it doesn't have any immunogenicity and the body cannot recognize it as an antigen. Then degradability. It's not just that the antigen can enter the body, but that it has to be able to be degraded by these substances or immune substances inside the human body. So we have the macrophages, the neutrophils, the dendritic cells, that they have two main functions: antigen presentation and antigen neutralization, antigen opsonization, and complement fixation. So long as when those substances acted on the antigen, they cannot be degraded, then therefore it is not antigenic. So you take example of someone swallowing a stone inside the human body. That stone cannot be degraded by the immune cells. but eventually it can be reduced in size and excreted in the pieces. So that's example of lack of degradability. Then the component of an antigen. The most important component is the epitope, which is just the binding site, or you can call it the receptor site to the antibodies. Number two, we have autoantigen. Autoantigen are antigen produced by the body. They are not coming from outside the body, they are within the body. So we can have some fragment of DNA, like prions, like plasmids, that they can be released into the body. You can have example of corneal tissues from the eyes. You can have example of other substances that they usually come from the body, and these may trigger an immune response, or they can be a very poor antigen. So they are termed autoantigen. Then we have alloantigens. These are antigens found in different members of the same species. Example, the red blood cell antigen A and B of the blood group. The antigen on the surface of the red blood group, they are antigens which we use them in differentiating different individuals with different blood type. That's why we have blood group A positive, B O negative, O positive. We use them in differentiating different category by blood antigen differentiation. Then we have heterophilous antigen. These are antigens that are identical antigens found in the surface of different species across reactive microbial antigens. So a particular microorganism may have the same antigen which different individual they can react to. It's like you have Salmonella typhi with serotype A and B. It comes from the same species, but they can use to trigger immune response in different individuals. So these are called the heterophiles, from the word hetero, different category of antigen. And we also have the adjuvant. Adjuvant are substances that are non-immunogenic alone, so they cannot trigger immune response alone. They require the presence of a haptin. So haptin is immunogenic, whereas adjuvant is not. So for them to collectively work together, you have to combine adjuvant and haptin. So they will become strong antigenic and immunogenic substances that can trigger immune response. So this is the end of the second topic. We are going to continue with the third topic. Thank you very much.